Here's your lock and your key. When the key fits specifically into that lock, it will open up that door, right? Giving you access to the room, or therefore an end product, right?
You keep moving there.
Why not?
Well, how should we write it in essay form?
In essay form? You can say that enzymes contain a specific binding site for specific substrates.
That's it?
So the substrate will fit specifically into the binding site, enabling a enzymatic reaction to occur.
So the substrate will fit.
It will fit specifically into the active site.
Into the
Active site. You know, the active site that contains the cofactors and coenzymes that give them the energy in order to do that.
okay, the substrate.
And so that will allow for a product to be produced. That's your Reader's Digest version of it, I guess.
Product.
Produced. Yes.
The substrate.
Okay? All right. Next one. Describe the endosymbiotic theory and give evidence that supports it. What is the endosymbiotic theory and basis? Long time ago, you had an anaerobic cell. Okay? Anaerobic meaning that it carried out its metabolic processes without oxygen, right? Anaerobic. Well, now you have this smaller aerobic cell. That smaller aerobic cell was engulfed by that larger anaerobic cell. Okay? A cell that is aerobic, how does it process its energy production? Through aerobic respiration, right? Aerobic respiration is going to yield, number one, more ATP and energy, right? So when that smaller cell has been engulfed into the larger cell, it's going to give a whole lot more energy to that larger cell than it was able to produce on its own. So therefore, it developed a symbiotic relationship with the new cell, meaning I'm going to produce more energy for you, and you are going to protect me from the outside. Okay? So, endosymbiotic relationship. So, that's the theory,
Where they both benefit.
where they both benefit from being together. The smaller cell benefits from the larger cell because it is now given an environment to which it can thrive and live and be protected from outside insults. The larger cell benefits from having that smaller cell in there because that smaller cell is producing more energy than it could produce on its own. All right? Now, look at your mitochondria, right? The mitochondria in your human cell produces a lot of energy. It produces a lot of ATP. It is also a organelle inside your cell that participates in aerobic respiration, right? Just like that prokaryotic cell from a long time ago. Now, how do we know that that theory is viable or could be true? has to support that with the evidence, right? Now, we know about prokaryotic cells because we're studying them, right? Prokaryotic cells, how do they divide? Do they divide independently, and do they divide through binary fission? Yes. Do your mitochondria do the same thing? Yes. Prokaryotic cells contain a 70S ribosome, right? Your mitochondria contain 70S ribosomes. Your prokaryotic cell has a single circular DNA or genome, right? Same thing with your mitochondria. It also contains a single circular DNA. Your mitochondria has a double membrane, right? So does the prokaryotic cell has a double membrane. So all these things that are similar between the two give us the evidence that sometime long ago, these prokaryotic cells were engulfed by larger anaerobic cells and developed this endosymbiotic relationship. That is your evidence that supports it. 70S ribosomes, single circular DNA, double membranes, they divide on their own through binary fission.
Okay, so at the end you said that gives us evidence
That this endosymbiotic theory is true or at least believable. There are some scientists that dispute this, and most of us look at the evidence and say, well, that makes sense. Okay. All right. Let's look at the metabolic process of fermentation. List the end products of this process. Fermentation is basically glycolysis with an extra step. Okay? I have a molecule of glucose, right? This molecule of glucose goes through glycolysis. And we know about glycolysis because we talked about it earlier. In glycolysis, you have 3 steps, right? You have investment, lysis, energy conservation. Now, at the end of that, you have 2-pyruvic acid, right? Now, you're also only going to get 2 ATP from this. So you're going to get a smaller amount of energy. 2 ATP are produced. Your pyruvic acid is produced, as well as some carbon dioxide, right? But if it was to go through a oxidation reduction reaction right there, at that point, which it does, it's going to produce a byproduct of an acid, such as lactic acid, acetic acid. In the case of yeast, what is it going to produce? What did Louis Pasteur do? Alcohol.
Wait,
Okay.
what was the other acid? There was lactic and what?
You got lactic acid, acetic acid. You can get propionic acid with the propionic bacteria, which gives you Swiss cheese, right? So the end product is going to be an acid. For example, lactic acid or acetic acid. If you ever took your nail polish off, you use some acetic acid, right? So your thank the bacteria for your nail polish result. I like to thank yeast for giving me wine and beer, some cheese.
Okay, so fermentation is glycolysis.
With an extra step.
Which the steps are investment, lysis.
Well, no, those are the steps before.
Okay, so we don't have to do that.
Yeah.
And it yields pyruvic acid, 2 ATP, carbon dioxygen, which then goes through the oxidation process.
Yes.
Can yield an acid, or
Yes.
with yeast produces an
Yes,
Okay, that works.
correct. And that is your exam. So does anybody have any, stuff inside the lab you need to take a look at? You're done. You're done. Are you done? Are you done? Did you do any tests on Wednesday?
I did.
So if you guys have time to go over to the lab right now, I'm gonna go over there, you can look at your stuff, take your pictures, and then get your evidence, and I'll put it inside the refrigerator. So, Jamie, are you done?
It was positive.
It was positive.
Yeah.
So you know both your bacteria.
Yeah, yeah, but
Well, the top part of that pretty much covers